So we've seen ways that chromosomes can be arranged in the cell and how chromosomes move around during the process of mitosis and meiosis. And this lecture is a little bit just on the broad principles of chromosomes and some patterns of chromosomes as they exist within the same species, sometimes between species. And my background illustration here, this is a plant called goat's beard. And it's one of many plants that are capable of having multiple copies of their genome. So they can have more than diploid number of chromosomes. In fact, that's very common in plants to have diploid, 2 copies, 4 copies, 6 copies, all kinds of variation in chromosome number. So we'll talk about kind of the genetic implications for that. What happens if you have an extra set of chromosomes? What happens if you have one extra chromosome or one chromosome too little relative to the rest of your species? What happens? So back to chromosomes again, we've seen this illustration a few times. This is the chromosome complement of a male human, diploid set of chromosomes, 23 pairs. And I don't have to tell you who this person is for you to look at this and have a pretty good guess that that's going to be a human. So looking across humans, again, this gets a little bit back to that term we introduced at the beginning, normal. This is a normal number of chromosomes, as in this is what most people have, 23 pairs of chromosomes. It is not unique, sorry, it's not widespread, unanimous across all humans, but most humans have 23 pairs of chromosomes. And so what you see here, not only the number of chromosomes, but the size, the distribution of genes. So if you looked at a portion of chromosome 2 here in this yellow region, across all the humans you would look at, you would probably find the same genes in the same order on chromosome 2 here. So there's a lot of very similar DNA across humans. Part of that is because we all have a relatively recent common ancestor as far as evolutionary time. Another reason, though, is that it's important for two people, when they get together to have a baby, to have compatible DNA. And that means that all these chromosomes, when they meet up between gametes, that they also can find their homologous chromosome and use that to perpetuate the species. So if you belong to the same species, then, you know, with some exceptions, certainly within mammals, this holds a little bit more true. Same chromosome number among humans, chromosomes that have the same genes in the same order. And we'll kind of see a little bit more as to why these are important. But it's important to be able to line up your genes the same way. So one of the chromosome concepts we're going to talk about is this idea of dosage. And this is a kind of a big concept in genetics. So genetic effects are not always just due to whether you have a wild type version of a gene or a mutant version of a gene. There can be lots of other ways to have different genetic effects. And one of those is through this concept of dosage. So this is basically a concept of balance. So dosage, one way to think about that when we use dosage in terms of taking medicine, you take a different dose depending on your body weight. So if you go to the hospital and you need to take some medication, they're going to learn about your body and give you the amount of medication that's appropriate for the size of body. When you're a child, you take small amounts of medicine to treat the pain for painkiller. As you get older, you take larger amounts of medicine, and that's just proportional to the number of cells that it's receiving, that's getting sent to. Same thing kind of with genes. I want to encourage you to think about dosage in terms of balance. So this is showing a set of gene products A, B, and C. They make proteins, and those proteins interact together. And the balance of A to B to C is actually pretty important in a lot of genetic interactions. So genes, I should say maybe protein interactions. So if you have an extra player in this arrangement, A, B, and C, then that extra copy on the right-hand side, the extra copy of gene B, just looking at one chromosome, this would be a diploid individual, but an extra amount of gene B messes up that interaction. So they're just sort of giving you a hypothetical here, not really spelling out why the interaction would be different, but hypothetically something changes in the situation where there's a double amount of B, and that causes something to develop differently in the development of this organism. So just kind of general principles for now. It kind of gets into biochemistry that I want to avoid getting into too much depth with why these are, but maybe a little bit of an analogy here. So if you think about gene dosage, you can think about baking or recipe. So if you're going to make some cookies, right, you need your eggs, your flour, your butter, and sugar and all these things. And if you're off a little bit, you could probably still make a pretty viable cookie. But if you went in and you just double the amount of flour, maybe you've done that by accident. You double the amount of flour and keep everything else the same, your cookie is going to be really dry, right? Double the amount of butter, keep everything else the same. You might get a better tasting cookie, but one that doesn't hold together well. And so one way that you could make a double amount of butter is if you double the butter, also double the flour, also the eggs, and every ingredient you doubled, that would actually keep the dosage the same. So it's this relative amount when we're talking about gene dosage that keeps the balance just right. And we'll actually see there's a case where you can, like I said on the opening slide, double your amount of chromosomes and still have a viable organism that has a balance of genetic information. Here's a dosage example from mammals. We talked about this situation where males inherit one X chromosome and females inherit two X chromosomes. This is widespread across mammals. We've seen other forms of sexual determination, sex determination. But this one is probably the most familiar, and we'll stick with this because it has a nice dosage connection. So if you've got a male human and a female human, so we got, sorry, hold on here. Dun, 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 dun. Sorry, friends. Patience. There we go. Unplug my notepad. There we go. All right. So maternal and paternal is referred to the mother and the father. So from mom, she gave an X and dad gave an X. And so this individual that got an X and an X would develop as a female. And that would make her very different from the situation in the male. So males get one X. And so inside of their cells, inside of their nucleus, they get one X chromosome. And females get two X chromosomes. And we've seen that there are genes on the X chromosome that are not always related to sex. They can just be something, we've seen some genes related to color perception, for example. And so a male having one set of genes for color perception and a female having two sets of genes for color perception could throw off some of those balance dosage arrangements. So mammals, their cells a long time back figured out how to fix that a little bit. So females get two copies, males get one copy, and in the female, one of those copies gets inactivated. So at some point during development, early on in development, one of the X chromosomes gets inactivated through a process that we'll see a little bit later in the course. And it's basically random which one of these chromosomes gets inactivated. So we've got mom's copy on top, dad's copy on the bottom. This cell has mom's copy inactivated, this one has dad's copy, and so on. Basically random. And it's early on in development. And once that shift happens where the chromosome is inactivated, the same chromosome stays inactivated. So we get interesting patterns. One of the ones that is maybe familiar to you is in cats. There's a kind of cat called a calico cat. And calico cats have this pigmentation pattern that's just sort of like kind of blotchy, random pattern. Another one is called tortoise shell on cats. And it's sort of asymmetrical, a little bit different than you'd expect on a patterning. And it turns out that the coloration on these cats, the orange patches and the black patches, are cells that descended from cells that had one version or the other of those X chromosomes turned off. So one of the X chromosomes being on produces black fur. And so these would be a population of cells that descended from an initial cell that turned on or kept on the black pigmentation X chromosome. And over here, there's a patch of cells that descended from the original cell that kept on the orange pigmentation X chromosome. And so what this means is that males have one X, and females, by inactivating one of their X, they also have one active X chromosome. So it's sort of like the normal dosage for an X chromosome in mammals is one copy of X. One thing you might have wondered, sometimes comes up on that question, is, wait, one more question. So if you have a calico cat, or if you've encountered a calico cat, I like to ask students, a lot of them have seen or maybe currently have a calico cat at their house, and I ask them, what sex is your cat? And most people say that their calico cat is female. So this is a feature that is exclusive to females. There's always exceptions, but almost always female cats with this calico or tortoiseshell appearance. All right, so you might wonder if there's some kind of adaptive disadvantage to having one of your X chromosomes inactivated. Let's say you're heterozygous for a gene on the X chromosome, and the X chromosome that's got the functional version of that gene gets inactivated. Well, what happens then? And the short answer is, I suppose it depends on the individual cell, but this is another version of the answer, is apparently the cells in the neighborhood can kind of make up for that. So the cells that that leave on the instructions for making color perception proteins, they can help out the cells that do not have the instructions for making color perception proteins. So it's either the patchiness of the cells at the color perception part of the eye, or cells can possibly transport their proteins across into neighboring cells. I have to look into that a little bit more. Anyway, so this is why a heterozygous woman, heterozygous for color blindness, is typically not actually colorblind at all. Okay, so general principles of dosage, now getting into some examples where we can watch the chromosomes end up being different in different cells, different organisms. And this is just an illustration showing basically the terminology we're going to use for some of these. We'll spend a little bit of time in the left-hand example called rearrangements. So a chromosome rearrangement, sometimes these are referred to as chromosomal mutations. A chromosome rearrangement is where part of the chromosome gets shifted around somehow. So the genes that are on the chromosome, big chunks of DNA containing genes, are sometimes duplicated, sometimes reversed, and so on. We'll see examples of that. And so in this case, this is a duplication. This is a portion of that labeled chromosome that has CDE genes up top, and now it becomes CDE, CDE. This is a duplication. Another type of chromosome mutation is called aneuploidy. And aneuploidy refers to an unequal number of chromosomes. So we'll see on the right-hand side here, polyploidy is actually a way to preserve dosage if every chromosome has the same, is increased in the same amount. So this is going from 2 copies of each chromosome to 3 copies. This is a so-called triploid individual. That's polyploidy, and a little bit different from polyploidy. Aneuploidy means an unequal number of chromosomes. So we have 2 and 2 and 3. Aneuploidy typically is adding 1 chromosome or removing 1 chromosome from the normal chromosome amount. So this is one called trisomy. That means 3 chromosomes here, and you could have a situation where you call this trisomy, then the number of the chromosome. All right, so this is just some terms that can talk about how the parts of a chromosome can be rearranged. They should be pretty familiar, and I don't want to dwell on them too much, but these are terms you probably have encountered in biology before. So inversion, switching the order that these genes exist in. Duplication, as we saw before, adding, repeating a part of the chromosome that's got 1 or more genes on it. A translocation, that's moving to a different part, sometimes within a chromosome, sometimes across chromosomes. And this one is a special case called reciprocal translocation, where the E and F from the gray chromosome is being swapped with the Q and R from the black chromosome. And then over here on the bottom right, this is deletion. Deletion in this case involves removing the E and F genes from that chromosome. So getting back to the question of dosage, how does dosage relate to these chromosomal rearrangements? This is just showing one chromosome at a time. This would be a diploid organism potentially. But as far as this one chromosome, adding or removing copies of genes would get you a difference in dosage for the genes that are affected. So let's maybe just take one of these at a time. So this inversion here, this takes the DEF region and flips it around. So it's going FED. The genes look backwards. But if you remember, of course, the DNA is anti-parallel, so it's got two strands, and it can be read either way. So if you take a gene and you flip it around, if you flip the whole thing, the coding region, the promoter, the terminator, then it's going to look the same to the cell going forward or backwards. So this actually doesn't really have an effect on the genetic information that's contained by that chromosome. And that would be no change to the dosage. So there's still a functional D, a functional E, and a functional F gene in this inversion example. Duplication, extra E, extra F. So that, hopefully you can appreciate, would change the dosage. Additional copies of E and F. Deletion, we'll jump down to that one. Removing copies of E and F, that would definitely change dosage. Going back to our recipe example, this would be extra flour and extra eggs, let's say. And then this would be, you forgot the butter. Big effects on the recipe. Or even if you're looking at a diploid organism, if you forgot half the butter or added 50% more eggs, those could still have pretty big effects. Translocation, even though these genes are on different chromosomes, they still have the same genetic information. So a translocation here, as long as nothing gets deleted, actually preserves the normal dosage. So dosage, we can look at the dosage in the cell that has the chromosomal mutation, or we could look at what happens to that chromosome as a result of its chromosomal rearrangement during meiosis, during mitosis. I always hesitate a little bit to show this slide because it is kind of involved and it's a rather special case situation. But what they're showing here is that this kind of, which this one again, it's an inversion, right? So we go CDE, shifts the position of the centromere a little bit. And in the process of meiosis, as these chromosomes line up with each other, they could recognize each other here and here, but then they'd be a little bit thrown in this region where C should be matching with C and E with E. And they suggest basically this could happen. So crossing over normally happens between chromosomes that are not rearranged like this one. But this is a possibility where during crossing over, they're trading parts that are not homologous because this D region is a little bit different than it usually is. So as a result of that, we get a pretty wide variety of chromosomes here. So we got the normal version on the top. We have one that got extra F and G as a result of that crossing over. We got one that got so extra F and G, but no A and B. This one got extra A and B, no F and G. And then the bottom one still has, as it says there, the normal number of genes, but it still has that inversion relative to the normal gene position. So if you're looking at meiosis, these 4 chromatids shown at the bottom here, each one of those is going to end up in a gamete. And a couple of those would be able to fertilize another gamete and have either no effect whatsoever or no effect in the next generation, but still kind of a problem. And then these 2 in the middle, those 2 chromatid versions, would have a dosage effect in the offspring of that cross. So meiosis can take an inversion or something like that that doesn't immediately change dosage, and it can generate a dosage problem as a result of meiosis. How about mitosis? Let's see if we can diagram this one. We left ourselves some space here. Maybe we can do it. All right, so in mitosis, do a little bit better on my other drawing pad. So in mitosis, if you remember, the chromosomes that are homologous do not line up next to each other. A, B, C, D, E, F, G. As a result of DNA synthesis, they would have identical halves. So that's the top chromosome. Bottom chromosome here goes A, B. I'll go ahead and write my letters backwards just like they do. C, D, E, F, G, A, B, E, D, C, F, G. So in mitosis, if you haven't caused a dosage issue immediately, then the rearranged chromosomes can actually keep going indefinitely in mitosis. As far as one cell dividing into two cells, this can still preserve the dosage. And as long as the cell doesn't go on to produce a gamete, it's kind of not going to have an effect on the organism. So nice reminder. I think one of the big differences, mitosis and meiosis, is that mitosis can kind of get around dosage problems that would otherwise arise in meiosis. I guess we say dosage problems. Maybe some of that dosage could be an interesting advantage if you generated some variation. All right, so I told you I didn't want to hover on this for too long. This is another illustration of how so this is translocation. This is another chromosomal rearrangement, how it can affect the resulting cells during meiosis. If you have a translocation, you can have one of these four-parted chromosome interactions when the chromosomes line up with their homologous versions. Normally be red with red, but this red one has a piece of blue on it. And this red one is entirely red. This blue one has a bit of red on it, and so on. So you've got two chromosomes that are kind of splitting their time between red and blue. And I'll just try to get ahead to the video that shows this. There are different ways that they can line up. So the machinery is used to doing this with two chromosomes in pairs, and pulling them apart at some point. And if there are four associating together, they get pulled apart a little bit differently. So a few ways in this can happen. We got N1 and N2 can go off together. Translocated 1 and translocated 2 can go off together. So going back to this, it's basically like we have four together, and two of these centromeres are going to get pulled to one side and two will get pulled to the other side. So this one can get you two normals in the same cell. This goes ahead and gets you a functioning normal gamete, translocated gamete with okay dosage, translocated gamete with okay dosage. These are the ones that are called viable. So everything in this column here has a normal number of all the genes that are represented. Everything else has either too many reds, too many blues, and so on. So a mismatch of the number of reds and the number of blues. Some genes are missing, some genes are in excess, and everything in here is a dosage problem. And they're labeled as non-viable. So even without telling you what the genes are, this is a big enough effect that it probably would cause the organism that inherits this not to survive. Here's a nice video. There we go, maybe. We'll see if this comes across on the recorded version. So this is just an animation showing that thing that we were just talking about, where you have four chromosomes that are translocated, and so they're kind of partly homologous for two different chromosomes at a time. And depending on how that shakes out when they separate during metaphase 1, anaphase 1, they go to different parts of the cell. And let's see, on the left-hand side here, we're getting too much pink on top and too much yellow on the bottom. Over How's the other one working out? The other one has, yeah, normal pink and yellow on top, and then the okay complement of pink and yellow at the bottom. All right. So chromosome rearrangements, just generally speaking, some of them immediately cause a dosage problem, and other ones, if they don't immediately cause a dosage problem, they'll probably cause a dosage problem for at least some of the cells that result from meiosis. So now moving on to chromosome number variation. I mentioned this term aneuploidy. Aneuploidy is a Greek construction of Greek words, and it starts from the root of euploidy. So ploid refers to chromosome number. We've talked about diploid and haploid. And euploid individuals, E-U-P-L-O-I-D, euploid individuals have sort of the right balance, the normal balance of chromosomes. And so you can have euploid numbers that are polyploid. So they're balanced, but they can be balanced as pairs, as threes, as fours, and as 1, 2, 3, 4, 5, 6, 8. 8 copies of each chromosome in this species. So this is a group of plants called astragalus, and each of these is a different species showing a different number of chromosomes. So they would have a common ancestor that probably had a diploid 2n equals 16, 8 types of chromosome, but variously along their long course of evolution, they might have caused, at some point generated triploid, tetraploid, octoploid versions of themselves, and those went off to become their own species. So everything gets its own name and maybe looks a little bit different from the other ones. So these are polyploid versions. This is something I mentioned is fairly common in plants, much less common in animals overall, although there's some animals that do have polyploids. And the other version of chromosome number variation is aneuploidy. And so that's an unequal number. That would be a dosage effect by having one extra or one less than the total chromosome number. That's what we usually encounter. So something you might be familiar with in humans is the situation called Down syndrome is 3 copies of chromosome 21. So trisomy 21, and we'll see the effects of that in a little bit. You can compare chromosomes across species, kind of illustrate again that for species to be compatible with each other, they need to have genes in the same place on all the chromosomes. And I don't really have an illustration for that, but maybe you could think about if you were getting, let's say you managed to breed a human and a mouse together, very adventurous of you. You might have a full set of human genes and a full set of mouse genes, enough to maybe make a viable offspring. But when it came time to rearrange those into gametes, the genes on the mouse chromosome 7 are spread throughout the human genome. So this is, a lot of these would be nearly identical genes, and then a whole bunch of them would be different enough so that we have a human version and a mouse version that have diverged quite a bit. But the colors, basically, if you can match the colors, the features found on human chromosome 7, I don't know if I'm seeing this just right. Maybe over here on chromosome 3, there's a little bit of that green. Chromosome 11, a little bit of that green. This is showing us the effects of the long time since the human and mouse common ancestor diverged. There's been a lot of rearrangement of the content of chromosomes. Interestingly, though, the X chromosome pretty intact. Okay, so we have different gene content, also different chromosome number. Mouse has 20 sets of chromosomes, we have 23. A little more similar set of organisms would be comparing human and chimpanzee. That's the H and the C here. And we share a common ancestor only in the few million of years ago. And humans and chimps have a lot more genetic information in common. So the similarity of the painting on the chromosomes is a similarity in gene content. And I don't know that a human chimp hybrid would be viable, but a lot better chance than a human mouse hybrid, I guess. So again, this kind of tracks back to the common ancestor of humans and chimps. And in the time since that, there have been some chromosomal rearrangements. if you have two species that can make viable offspring, sometimes they're just a little bit under the ability to make, say, a viable species by crossing between them. So a very familiar example, hopefully, is the cross between a horse and a donkey, which generates a mule. And that is a pretty reliable result of this cross. So you take half of the genetics of a horse, so a haploid horse amount mixes with a haploid donkey amount, gets you a viable mule in the first generation. But you probably also have heard that mules are sterile, and this is a nice illustration of why that is. So if the mule went on to produce sperm and egg cells, those would get kind of a cherry-picked amount of some of its horse chromosomes, and some of the A on here is ass instead of donkey, some of the donkey chromosomes, and they're not always the same. So in this case, we've got some horse and donkey chromosomes mixed in here. And this box here says we've got some donkey chromosomes that aren't necessarily going to match up very well with these horse chromosomes. They've got different numbers on them. They've got substantially different gene content. It's a little bit of a wild card as far as finding out what's going to successfully pair up during meiosis, and pretty low chance that this mule would produce, pretty much an astronomically low chance that the mule would produce an egg cell that looks just like a horse egg, or a sperm cell that looks just like a donkey sperm cell, because there's so much of that, as we saw in meiosis, randomizing of the content of these chromosomes, crossing over, pairing, independent assortment, all this stuff. All right, so chromosomes are a big reason why hybrids are not fertile. Okay, so aneuploidy, I mentioned the term. This is an illustration of one of the ways, kind of the big way when aneuploids can form. This is a process called non-disjunction, which is a fancy word, just means that the chromosomes have not split in the normal fashion at some point during meiosis. So if we're just targeting this one chromosome, big red chromosome, has an okay normal split in meiosis 1, but in meiosis 2, fails to divide into two of these cells equally, and one of them gets 2 copies of chromosome red. One of them gets no copies of chromosome red. And if they get mated with a cell that's got chromosome blue, so the homologous version of red, these normal gametes become diploid. That's fine. And these gametes, when fertilized, become either triploid with 3 copies, or I guess it'd be, sorry, I shouldn't say triploid. The whole thing would not be triploid. It would just be trisomic. Let's use the right term there. So 3 copies of this chromosome trisomic, 1 copy of this chromosome monosomic on an organism that's otherwise diploid. So I mentioned Down syndrome and trisomy 21. And these are illustrating the chromosome complements of aneuploids that are known to be possible in humans. And these are, they've got syndromes in their name. They're basically collections of features. That's what a syndrome is. So collections of phenotypes, so appearance, sometimes genetic predisposition to disease. And these are some of the ones that we encounter in humans. So Down syndrome is pretty well known, pretty widespread as these go. Other ones, Klinefelter syndrome, Turner syndrome. Chromosome 21 is trisomic in Down syndrome. These other 2 involve the X chromosome. So Klinefelter, 2 X's and a Y. And this one, so-called XO, is X and nothing else. So this question here, why are these viable, whereas a lot of other forms of aneuploidy are not viable? Why don't we see trisomy of chromosome 2, for example? Well, the fact is that chromosome 2 has a lot of genes on it. And when you are trisomic, when you've got aneuploidy for a chromosome, that means that you've got extra or missing copies of all the genes on that chromosome. And the dosage effect of a big chromosome like chromosome 2 would be too much, and that is non-viable. Chromosome 21, on the other hand, is pretty small. And so having 3 copies of chromosome 21 causes you to have an unequal dosage, abnormal dosage of the genes there, but not as many genes. So small chromosome means relatively small effect. The question with the X chromosome here goes back to what we saw earlier about X inactivation. So if you have two X's and a Y, one of those X can be inactivated, and this individual will have mostly development like a male. So one functional X and a Y. Same thing here, missing an X is pretty similar to having an X inactivated. So it is just one intact X that has to do all the heavy lifting for all the cells. So they're not without any effects. They have names to them, so they're something that a physician might be able to diagnose without looking at chromosomes, but they're definitely viable and able to be functioning humans in their own right. So if you're curious about what it is that contributes to Down syndrome, Down syndrome is a collection of features, some with, as I said, with phenotype and some with health. And they're kind of common across individuals that have Down syndrome. So having an extra copy of 21 has a sort of predictable effect. And so these are the genes involved. These are chromosomes that are located, genes that are located on chromosome 21. And on the left-hand column there, it tells you what part of the Down syndrome phenotype they contribute to. So lots of active study in this department, and probably learn some interesting new things as we go. Another thing you might have heard is that in human pregnancies, as the mother gets older, the chance of something like aneuploidy, Down syndrome in the children increases with time. So the genetic mechanisms, the molecular mechanisms that assort the chromosomes into gametes, those break down a little bit over time. So with the age of the mother, the chance of this goes up. So this is a reason, if you are a person 40 years old or older and you're planning to have children or on your way to having children, you'll get probably some additional genetic testing as part of that. So lots we still understanding and probably improving everybody's outlooks as we're getting a better handle on these.